Welcome to MOOC NPTEL course on interactomics. Today we are going to talk about basics of surface plasmon resonance SPR with focus on surface chemistry. The phenomenon of surface plasmon resonance has found its way into practical applications in sensitive detectors capable of detecting biomolecular interactions, performing thermodynamic studies as well as quantitative analysis of biomolecules. It has become a key biosensing technology in the areas of biological research and medical sciences because of its application in real time, label free and non-invasive nature. The SPR instrument is comprised of optical detection system, microfluidics and sensor chip surface. The biomolecular interaction takes place at the sensor surface, which plays a crucial role in performance of a biosensor and the quality of data retrieved. In today's lecture, we have invited Dr. Srinivas from GE Healthcare. Welcome, Dr. Srinivas. In NPTEL MOOC course on interactomics, Dr. Srinivas will elaborate on some of the features of SPR sensor and their surface chemistries, and he will talk to us how they can be utilized in studying biomolecular interactions.
Thank you for the invitation.
Surface plasmon resonance, or SPR, is used to monitor binding events between molecules ranging from ions to viruses. This technique allows you to observe binding and measure kinetics, affinity, specificity, and concentration without any need for labels. BIACOR T200 is designed for ease of use and exceptional sensitivity. In BIACOR systems, molecular interactions are monitored on a removable sensor chip by the surface plasmon resonance detector. Samples and reagents are held in removable racks and are delivered to the chip by a microfluidic system that uses very low volumes of sample, down to a few microliters. In addition, the microfluidic system supplies the sensor chip with buffer from the buffer bottles and delivers waste liquid to the waste bottle. The operation of the instrument and the data collection and evaluation is handled by intuitive software. Let us show you how it works. A glass slide coated with a thin gold film creates the sensor surface. For most applications, a dextran matrix covering the gold film acts as a substrate to which molecules can be attached and provides a hydrophilic environment for the interaction. Other matrices can be used to attach specific types of molecules. The specificity of the surface is determined by the nature of the molecule attached to it. So one binding partner is attached to the surface of a sensor chip, and the other is injected in a continuous flow of solution. Whatever the nature of the molecules involved, we call the attached interacting partner the ligand, and the partner in solution the analyte. BIACOR uses the phenomenon of surface plasmon resonance to detect biomolecular interactions as they happen. SPR causes a reduction in the intensity of light reflected at a specific angle from the glass side of the sensor surface. As molecules bind to the sensor surface, the refractive index close to the surface changes, altering the angle of minimum reflected intensity. The change in SPR angle is proportional to the mass of material bound. The sensor surface, the microfluidic system, and the SPR detection unit work together to measure biomolecular interactions. The result from the detection of change in refractive index is displayed as a sensorgram, where the binding response on the y-axis is plotted against time in the x-axis. Since light does not penetrate the sample, analyses can be performed on colored, turbid, or opaque samples. From studying the shape of the sensorgram produced, binding yes or no, specificity, affinity, kinetics, and active binding concentration can be determined. The sensorgram provides real-time information about the entire interaction. This means that in a single SPR experiment, you have now obtained a wealth of information about your binding, which helps you understand the dynamics of the interaction or to quantify your analyte. And all of this without using labels.
Before we start with other sessions, I think we will revise a little bit of introductory slides on the surface plasmon resonance. A Biacore or a surface plasmon resonance systems are a label-free technology. These systems are generally used to monitor molecular binding events in real time. So, basically an experiment starts with an immobilization of a ligand on the sensor surface, and an analyte is passed over in a solution and which will bind to the ligand. So, here there are two different molecules, one of them is a ligand and analyte, and the interaction is recorded in a real time, and the data that is output on the surface in a sensogram, you see the binding events, which is also called as an association event, and there is also a dissociation event, and once the association event is also called as on rate, and the dissociation is also called as off rate. So, when you do one on the other, the association rate divided by the dissociation rate gives you the equilibrium Kd or a capital K capital D. So, this is the output that we get from a Biacore experiment. The sensogram that is generated from the Biacore or a surface plasmon resonance experiments tend to give us different informations. So, they tend to tell us whether there is binding. So, there will be yes no binding, and once there is binding, we will come to know if there is a specific binding or a non-specific binding. Once we have determined the binding, then we will understand how strong the binding is or how weak the binding is. So, once we have determined this part, then probably we will be able to understand how fast or how slow the molecules are interacting. And once we have come through this stage, I think it is very easy for us also to determine what is the exact concentration or how much amount of the analyte that is binding. From this kind of an experimentation, the comprehensive information that one will generate are detect, that means yes and no binding, identify the binding partners, identify the specific binding, and characterize the binding by different events like affinity, which is how tight or how weak the binding is, kinetics, how fast or how slow the binding is, concentration, which is how much amount of our analyte is bound to the ligand, and thermodynamics, how kinetic changes over a function of temperature. So, these are the different comprehensive information that is generated from Biacore experiments. Surface plasmon resonance will help us to understand a wide range of biomolecular interactions, be it be proteins, nucleic acids, lipids, membrane associated molecules, carbohydrates, low molecular weight compounds. The molecular weight of those compounds can be below 200 Daltons, whole cells, viruses, bacteria. All of these molecular interaction process can be understood very well using surface plasmon resonance experiments. So, the cornerstones of any SPR system or a Biacore system is a detection system and a chip system, a microfluidic system, and the software which integrates all of these and generates data. So, in our next sessions and next couple of other sessions, we will anyway understand details about sensor chips and the different sensor chips available, that the SPR detection system depends upon the refractive index and also depend upon the surface concentration and temperature. The interactions happen in a miniatured system called microfluidics, which are the liquid handling part of the system, which contains very low volume of reagents, and they are integrated and automated liquid handling systems where the flow cells are actually situated and the actual interaction happens and recorded by the system. There could be many ways of orientation of the microfluidics and the flow cells. It could be a parallel way, a serial way, or a two cells at a time, and different systems can have a different way of the fluidics and also different orientations. Control software provides different ways of running the system. So, there are events in which we could observe them manually. There are application wizards where we can set up experiments very easily, or we could write our own method and record an experiment. Apart from that, there is also a evaluation software that evaluates the data, and that data evaluation happens by use of algorithms which fit the data and will give out a lot of other parameters for any kinetic event. In this session, we will understand the different kind of sensor surfaces and the use of them in immobilization of a ligand. If we know from the basics and from other sessions, we have understand about the surface preparation, direct immobilization or by capture methods. So, direct immobilization is an covalent immobilization which is a permanent immobilization, and in this process we immobilize our ligand, the choice, and on a surface permanently. Other way of doing it would be a capture, and so in order to give a different sensors available or different surfaces that are available, and we go into each one of them a little bit detail to understand what they are and what is the major utility of them, and how do we design an assay based on these chips. So, the various chips that are available for any SPR experiment, to start with, they are named as CM5 chips, CM4 chips, CM3, C1, streptavidin or an SA chip, NTA chip, L1, HPA, and AU or a plain gold surface chip. Each of these chips have a place for an adapter, and this adapter actually holds the gold, and that gold place is where the immobilization happens, and the adapter actually pushes the gold surface into the SPR system where actual immobilization happens. If you look at a particular gold surface on a chip, it contains a layer of dextran, and that dextran provides or supports the interaction or immobilization of your ligand on the chip. The dextran is glued to the gold, and the gold is present in form of a cassette, and that cassette entirely is called as chip. So, looking into this gold surface, the major component attached to the gold surface is the dextran, and it is very important for us to understand why dextran, because dextran is hydrophilic, dextran is flexible, dextran resembles a solution or a aqueous solution, 2% aqueous solution, and it has a very low non-specific binding and a very high binding capacity, easy to activate for covalent coupling, and withstands extensive regeneration. Now, let us go into the available different chips. To start with, we will do the first chip, and universally accepted used chip is a sensor chip CM5. CM stands for carboxymethyl, 5 is certain number in which it shows the amount of branching. So, if you look at this particular slide, the chip actually has some kind of a glue, or gold has a glue. The glue actually attaches the dextran to the gold, and on the surface of the dextran you see those small areas which are called carboxymethyl groups, which are getting activated, and the activation of the carboxymethyl groups helps us to immobilize our ligand by amine coupling or a thiol coupling or a aldehyde coupling. The reactive side chains on the basic amino acids or amines are generally used to couple to the surface by amine coupling. Thiols are activated and then used to immobilize on the surface in a thiol coupling, whereas the carbohydrates that are attached on the glycoproteins are used to do an immobilization, and that process is an aldehyde coupling. And as I just said, CM5 is a very versatile chip, and any of these process or any of these methods can be used to immobilize on a CM5 chip. Coming to another sensor chip called as CM4. CM4 has a little less branching and a low charge, which is less negatively charged, and this particular chip surface is good for low immobilizations or low Rmax immobilizations, especially cases where you are looking at interactions involving biosimilars. Coming to another chip surface, which is called CM3, another way of branching of the sensor, the carboxymethyl groups on the sensor. Here again the matrix is less or little shorter than CM4, also convenient for low immobilizations and generally good with cell or viruses immobilization or multi-component complexes. So, coming to another sensor chip is a C1 sensor chip. C1 does not contain any dextran here, and it is a flat bed of carboxymethylated surface, very good with positively charged proteins, and can be used again with cells and viruses for a total immobilization of cell viruses, and been used by many available in the literature for immobilization of bacterial cells and mammalian cells, and useful for studying interactions at cellular level. The CM surfaces are very good with non-specific binding, which means they have a very little non-specific binding when they are used with culture mediums, when they are used with cell lysates, when they are actually used with serum in a diluted way and in an undiluted way. Also, these days people use these sensor chips for immobilization of cells, bacteria and viruses, and also for the passage of very complex analytes like blood. So, another variety or of a chip surface is a streptavidin. Here, this is also a CM5 chip, which is pre-immobilized with streptavidin molecules, and these streptavidin molecules have a very high binding affinity to biotin, and these streptavidin molecules bind to biotinylated proteins, peptides, oligos, DNA, RNA, and will help to immobilize these molecules in any interaction process. So, ideally literature is available for biotinylated DNA fragments, nucleic acid interactions, and other things like peptides have been immobilized on this surface. Another variety of chip or a different chip surface is NTA, and this is a complex molecule that is immobilized on the surface of the chip. It is pre-immobilized, and this molecule has an affinity for hexahistidine molecules or tags that are present either at the C terminal or N terminal of a protein. So, proteins when they are recombinantly expressed with these tags have been used to be captured on NTA surfaces for interactions with other analytes, either proteins or complex molecules. These histidine tagged molecules bind to NTA when the NTA is charged with nickel solutions or copper solutions. And these molecules generally help to control the steric orientation of the ligand for optimal exposure of your ligand. Another very important and a variety of chip surface is HPA surfaces are flat hydrophobic surfaces, and these surfaces are useful for membrane associated interactions. Receptors generally are anchored into a membrane like environments, and then they are useful for interaction with analytes. Generally, the lipid monolayers interacting with membrane binding biomolecules kind of studies are made on these surfaces. L1, another surface sensor chip which is also popularly used for studying liposomes, lipid bilayer kind of a molecules, and carboxymethylated dextran is modified with lipophilic substances. So, another surface is a plain gold surface where there will not be any dextran, and these surfaces are very good with chemists where they can do their own chemistries and useful for interaction. Nowadays, there is also something called the plain gold kits that are available, and these kits have a portable gold surfaces. The portable gold surfaces can be taken over into a laboratory, and you can do the required immobilization of the molecule and can be inserted into the chip cassette and then inserted into the Biacore system for studying the interaction. The summary of the different chips as we just went through, CM5, CM4, CM3, C1, SA, NTA, HPA, L1 are the different kinds of chips that are available for studying the different interactions. So, it is very important for us to identify what are the functional groups that are present on our ligand and how well we could immobilize our molecule on the different variety of a chip. We are coming to end of this session. So, with this session we have a great understanding of the various chips and the various chemistries that are available. With this, we will be able to identify whether we are going for a covalent coupling or a capture method. Once we immobilize our molecule, we will go ahead with our regular Biacore experimentation, and this particular session helps us to identify the right chip for our experiments.
Thank you, Dr. Srinivas, for this lecture. So, let us continue our discussion on surface plasmon resonance assays and experimental design in next class. Thank you.